So I would like to welcome you all to another klogW. Today we are hosting Dr. Erwin Frey, an APS fellow. There will be quite an introduction after I present our division slides. So I wanted to remind everyone that the deadline for the abstract for Global Summit is just 2 days away. So don't forget to submit to DSNP. The division additionally supports several travel awards for early career students. So if a postdoc or a graduate student has submitted to DSNP and they would like to participate in the competition for awards, they can request that. There's also a t-shirt design competition that's coming up, and they will all provide a travel award for the students and postdocs. Also, don't forget about the image of the month, where we highlight our research publications. If you go to our Engage page, you can find the external links and image of the month. Additionally, you can follow us on LinkedIn, Twitter, and Blue Sky. If you would like to volunteer for the division, we have a Google form, and we always welcome volunteers. I would like to go over the Zoom etiquette. Due to security issues, APS has decided to limit the attendance interaction with the host on Q&A. If you have any clarifying questions, please put them in the Q&A and chat box, and one of the hosts will interrupt the speaker and ask the questions. Save your other questions towards the end of the talk. After the talk, you can post your questions to Q&A. The hosts are able to unmute you and allow you to speak to the speaker directly. I would like to hand it over to my colleague to introduce Dr. Frey.
It's a great pleasure for us at the klogW series to host Professor Erwin Frey. Professor Frey is a theoretical physicist, a distinguished theoretical physicist who is chair of statistical and biological physics at LMU in Munich. He's made contributions to a very broad variety of problems at the interface of physics and biology. So there's a number of foundational papers from him on developing, the fundamental theory of semi-flexible and stiff polymers, applying them to understand the mechanics of very biologically relevant systems such as filaments in the cytoskeletal network, the mechanics of these things. Another very broad theme is the statistical physics of evolutionary systems and pattern formation in larger scales in biology. And as his page describes it, one of his central themes is identifying and characterizing universal principles and mechanisms underlying living or lifelike systems. It's a great pleasure to host him for us. This is his topic will be on the latter sets of things, and the title is Emergence and Self-Organization in Biological Systems. Again, he welcomes clarifying questions through the talk, so please feel free to put them in the Q&A, and one of us will make him aware of them. Welcome, and your stage now. Thank you.
Yeah, thanks a lot for your kind introduction and for inviting me here to give this presentation. It's a great honor for me, so let me pick the slides. I think that's it now. So here we go. Hope you can see it all well. Okay. As said, this will be a kind of broad overview of my research interests in emergence and self-organization in biological systems. And let me start with something quite general, namely that these systems are all far from thermodynamic equilibrium because there is one basic energy current of living systems, and this basic energy current is ATP hydrolysis. So you see here a transition from an inactive ADP state to an active ATP state. And this is really at the heart of many of the things I'm doing. One example is active matter theory, where you have molecular motors driving the relative motion of filaments with respect to each other, or you have molecular motors running along a microtubule, and you ask yourself about how is intercellular transport regulated. On the other side, this chemical energy of ATP hydrolysis is also used just for chemistry. And what I mean by that is that ATP hydrolysis is used to change the affinity of proteins to different compartments in the cell, say to the cytoskeleton, to the nucleus, to the membrane, and to other internal structures that you might find in the cell. And via that, what you do is you break detailed balance. And this broken detailed balance is at the heart of what I'm going to say. Here are two examples that I find particularly interesting, and they ask different questions. One is the self-assembly of complex biomolecular structures, like those cytoskeletal filaments, the ribosome, the flagellar motor, carboxysomes, viruses. And their main question is, since you're far from equilibrium, you want to know how long does it take for such structures to form. So you ask a question quite akin to computer science, where you discuss computational complexity. Here you ask about the time complexity, so how long it takes to form a structure given its size. The main theme for today will be protein pattern formation. And there I want to allude to you how this ATP cycle is contributing to the self-organization of patterns in a cellular context and how that guides biological function. Allow me to start with a quote from philosophy. It's a quote from Immanuel Kant's very famous contribution to philosophy. It's the critique of the practical reason. And he says that there are two things that fill his mind with awe and reverence the more he reflects upon it. And what he mentions is things, of course, I find also fascinating, which is starry heavens and the moral law within me. But let me twist it a bit in a way that fits better to me in my research. So I would say what fills me with awe and reverence is the living nature around me and the moral law within me. And to show you why I'm so much in awe by biology and living systems, I'll show you here a few examples, movies I got from colleagues, from collaborations that we had on different pattern forming systems. So the first movie that you see is a yeast cell, and there are proteins. They are marked by some fluorescent dye, and what you see is from an initial random configuration of these proteins, there's this polar cap forming on top of the yeast cell, and as you see in the cartoon, this polar cap that forms recruits actin filaments that form behind this polar cap, and then those actin filaments, they push out the membrane such to get budding, and this is the creation of a new offspring, a new yeast cell. So you see, pattern formation here has a biological function. The function is reproduction. Very similar, this is now a different type of cell. This is an E. coli cell, so a bacterium. There you have oscillation of these proteins also on the membrane between the two cell poles from the right to the left and back and forth. And now as you oscillate back and forth, what you have is you generate a density gradient such that you have minimum density in the middle of the protein and highest density at the two ends. And that density gradient is again read off by a polymer structure, the so-called FTSZ polymers. They form a ring, and this ring then contracts the E. coli cell, creating two daughter cells. Yet another example couples pattern formation with morphology. So as you see here, there's a starfish oocyte from a collaboration with Nikta Fakhri from MIT. And there you see, as the movie loops, there is a wave of chemical patterns around the starfish oocyte. And with the chemical patterns, you have the deformation of the oocyte. So you have a beautiful combination of non-equilibrium physics with the differential geometry of the confinement they are contained in. And finally, this is data from Petra Schwille's lab. This here is not a cell. It looks like a cell, right? It's moving in a cell-like fashion, crawling on a substrate. What it actually is, it's a vesicle, so just an empty bag of lipid molecules. And it's driven by the interaction with a pattern that you see here in green, kind of foam-like, mesh-like pattern. And this foam-like pattern is constructed from the same proteins that you saw giving rise to the pole-to-pole oscillations in the E. coli cell. So you have cell-like motion, which is one of the basic issues you need to figure out in order to move towards creating or constructing a synthetic cell. Now, I haven't shown you these beautiful movies. Let's go into detail and try to understand why these patterns emerge and how this is linked to interesting physics. So here again, you see the oscillation of the min proteins between the cell poles, and behind the scenes, there is this reaction network. So what you have here is two compartments. You have the membrane, which is a two-dimensional surface, and you have the cytosol, which is a three-dimensional bulk. And the main player here is a so-called ATPase, so a protein that switches its affinity to the membrane by going from the inactive ADP state to the active ATP state. As you go from this blue to the green state, the min-D protein becomes binding competent. So it's able to bind to the membrane. And once it's bound to the membrane, then there's a nonlinear feedback process kicked off that recruits more and more of those min proteins to the membrane. So transferring protein mass of min-D from the cytosol, from the bulk to the surface. If that would be it, then the whole surface would just be covered with those proteins and you would not see any kind of periodic pattern on the system. So you need an antagonist, and this antagonist is the min-E protein that you see here. It's also recruited to the membrane by the min-D, and then they form a dimer. And in this dimeric state, the min-E is hydrolyzing the min-D, so switching it off to the inactive state, and then both fall off from the membrane, go back to the cytosol, and you have the cyclic flux that you see here, a cyclic flux between states and a cyclic flux between membrane and cytosol. And that's a very hallmark of a non-equilibrium system, right? Because detailed balance would not allow such kind of cyclic currents. They are due to the fact that you have energy input in the transition from the min-D ADP to the min-ATP state. So that's the basic biochemistry, and from that you then have to construct a theory. But before I go there, just let me phrase and frame what kind of questions one would like to ask in studying these systems from the perspective of theory and or biology. So one is, of course, the relevance of the reaction network. And that's quite an important question because we all in statistical physics are trained in a way to say that details don't really matter and they can be coarse-grained away, and what really matters is just some basic features of the underlying reaction network. That's kind of true in those systems, but some of the features really very much depend on the detailed molecular nature of these reaction networks. And to spoil the story a bit, you need some kind of modules in the network that are responsible for different aspects of the pattern, like the time scale, the length scale, the robustness, etc. Another important question is, can those patterns actually sense the cell geometry? So as you see here on the right, these are data from K.S. Decker's group. where E. coli cells have been compressed to different cell sizes and aspect ratios. And obviously, as you see from the movies, the pattern depends on the aspect ratio. These are nonlinear systems, so it's not like in the question, can you hear the shape of the drum, where you select eigenmodes by the boundary conditions. It's a more subtle process where the interaction between the patterns that you have on the periphery, on the membrane, and the penetration of those patterns into the bulk that allows those nonlinear systems, those non-equilibrium systems, to sense the geometry they are contained in. And last but not least, and this is of course at my heart very much, is the question of, are there some general principles in these pattern-forming systems, and maybe even some new laws that one can discover studying these non-equilibrium pattern-forming systems. So let me start with the latter one. So here is again our yeast cell. What you can do is you can encode all the complex biochemistry into a reaction network. You use finite element simulations, and here we go. You reproduce the phenomena that you see in nature. And that's important and that's fine to have that. But let me now change gears and ask, how simple can you make it to still have the essence of the pattern-forming system in as few components and few species as possible system that you can imagine? Well, the simplest system you can imagine is one protein, right? And this one protein, let's say, is just in two states. One state is bound to the membrane, and the other one is freely diffusing in the cytosol. So just two components, one species. And once you have that, you can say, well, now let's write down a conceptual model that just reflects that idea. And this model has been written down a long time ago by Alan Turing in his 1952 paper. It's one of the classics of pattern-forming system. You have M and C, the two concentrations, F and G, the two functions encoding any kind of complex reactions. And the last terms that you see there are just encoding diffusion. So that equation has been very instrumental in kicking off the field of pattern formation, and you might have heard about Turing patterns, but it's not the proper model for what I have in mind, what we have in mind when we think about pattern formation in biology. And the reason is that it doesn't respect a conservation law, namely the very basic conservation law of mass conservation, that the number of proteins, M plus C, has to be a constant. And for that to be the case, what you have to replace the function G with is just minus the function F. So the two reactions in the two equations need to be equal and opposite, such that if you add them up, you cancel out the reaction terms. That seems like a really minor modification, but it has an important implication, and I hope I'm able to allude to what the important implications are. First, let's just add the two equations. And as you add the two equations, you can write them in a form which might remind you of the Kahn-Hiller model, or model B, in the classification scheme of Halperin and Holmberg. So you have the change in density, total density is a constant, the kinetic coefficient, times the gradient of a potential. And this potential eta in the thermodynamic case would be just the derivative of a free energy that you construct for a liquid system or for an Ising model. This is not correct here. It looks like it, but the object eta here is not a potential in the thermodynamic sense. It's a dynamic variable on its own right. So if I combine the two equations to write an equation for eta, what you do get is another dynamic equation for what we call the mass redistribution potential that's given by a diffusion term and also by a reaction term. And now let's us explore of what the consequences of such a coupled dynamics
May I interrupt? Sorry.
Oh, please do.
There's a question which I'll just read out for the moment. Why should protein mass be conserved if the cell is growing? Then perhaps the mass should be increasing.
Yeah, so if you look on the time scale of pattern formation in the cell, which is typically, sub minutes, proteins are neither degraded nor produced. So to a very, very good approximation, protein number is constant. Of course, it's changing slightly, but that's not the point. We need to take the limit series of constant value, and then changing it slowly will then just act as an adiabatic perturbation by which you change the behavior I'm talking about. Okay, so now let's make things simple, as simple as possible. So let's first look at a well-mixed compartment where reactions only take place. And let's say the reactions, the function f, contains two terms. One is attachment from the cytosol to the membrane, A of m, and the other one is detachment from the membrane to the cytosol. If I want to know about the steady state, then I just have to answer the question, when is that function f that I have there 0, because then dynamics is stationary. So what you plot is you plot the dynamics in phase space, which is the axis given by concentration in the cytosol and on the membrane. And the first characteristic function that you plot is the null line, that is the function f to 0, which is the set of all equilibrium points. And now you already see the difference. If you were to have a second function, like in the Turing case, there would be another line that you see, and this another line would cut the line you see at one definite point, and that would be the fixed point at which the system has to be. Instead, in our case, the second condition is given by mass conservation, so m plus c is n, and that defines a simplex. So if the system is well mixed, the dynamics has to remain on that simplex because of mass conservation, and then the fixed point is on that simplex. But now imagine by tuning the protein concentration by gene regulation, or as we just talked about, by the cells growing or the cells regulating the protein content, you change this number n, the total protein density. So you see the first key feature of mass-conserving reaction diffusion systems, namely that the density is a control parameter. And as we will see later, it's not just a global control parameter, it's actually a dynamic control parameter in a spatially extended setting, because density varies with space and with time. And that gives rise to the interesting interplay of dynamics that we have. So now that we have understood the well-mixed case, next go to the next simplest case where you have two compartments, which may reflect one and the other pole of your E. coli cell. And let's say we now have those two compartments be coupled by diffusion. And let's focus on them for the moment just by diffusion that happens through the cytosol, because that's the faster diffusion path. Just remember, diffusion coefficients in the cytosol is something like 10 micrometers squared per second, whereas on the membrane, because this is a lipid bilayer, it's only about 0.1 up to 1 micrometer squared per second. So now let's look at this phase space representation and try to understand graphically of what happens. Let's start at a particular point where the density is the same, equal to n bar, on the left and on the right-hand side. That's the gray spot that you see. And now let's assume that I perturb the system such that I increase the density on the right and decrease it on the left. And let's ask what happens now. So you now think of those two systems as isolated in two different spaces of mass conservation. And that means reactions will push you towards the respective fixed points that you have in these mass conserving planes, which are the black dots that you see. Well, but now something happened, right? Now you saw that the gradient in cytosolic density between the left and the right compartment even got larger, right? It got increased. And that means by Fick's law that there will be diffusive current from the left to the right, and this diffusive current then gives rise to an even higher surplus of density on the right, so you increase the difference in densities that you started with. Well, that just means you have a positive feedback loop that you generate, and any kind of deviation from homogeneity will be amplified, and that gives rise to pattern formation. And the criterion can be put very simply, because when you follow the argument, if I were to reverse the slope of my nullcline, I would get that the system relaxes back to equilibrium. So the criterion for pattern formation is simply that the slope of the line of equilibrium is negative. And then you have now a geometric classification of the emergence of pattern formation. And then in the end, if the system relaxes from the pattern forming state into a steady state, you will get a polar configuration between the two states, and I'll come to that in a second. I made things a bit simpler than they actually are. There's also cytosolic diffusion, and then the argument is pretty similar, and the argument tells you that you will have to have a slope that's steeper than a surface or line here that has a slope which is given by the ratio of the two diffusion coefficients, the one on the membrane and the one in the cytosol. So the basic lesson to be learned from that is the general mechanism for protein pattern formation is a mass redistribution instability, and it's basically the mechanism underlying polarity in yeast cells. So now let me show you how this unfolds in a computer simulation. What we see here is a simultaneous representation of what's going on in the phase space. That's on the right, C and M. And on the left, we have a representation in physical space, where here on the top, you have the membrane density as a function of space between left and right boundary. And on the bottom, we have the cytosolic density also as a function of space from left to right boundary. And as you see, there's small perturbations to the initial state, and we're now going to watch how the instability unfolds and what happens in the long run. So first, you get patterns with a periodicity given by a certain length scale. That's defined by the dispersion relation that you can calculate from linear stability analysis. And then on this final attractor of dynamics, there's still some dynamics going on, as you saw, namely there's coarsening dynamics. And that's quite akin to what one knows about Ostwald ripening in liquid-liquid phase separation, where you also have initial pattern formation by spinodal decomposition and coarsening by Ostwald ripening, giving rise to a meniscus, here giving rise to a polar pattern. You can also make the analogy even closer, though it's not the same. This is a non-equilibrium system, so you cannot really do thermodynamics here, but the constructions are very similar. So for example, there's a steady state, and the steady state is characterized by a flux balance. This line here in blue is the line where all the diffusive fluxes balance. That must be the case for systems to be in non-equilibrium steady state. And on top, you now have three hallmark points in phase space, each of them cutting the nullcline, and they correspond to the plateau of the pattern on the left, to the interface to, and to the plateau of the pattern on the right-hand side. You also have a condition of reaction balance, and that basically means that the region that you see now, so this shaded area or this area here, and this area there, roughly speaking, are of same size, such that chemical reactions in the system balance, and that's quite analogous to what one knows about Maxwell's construction. So if you take that and you make a construction, you can, out of this graphical construction, come up with a phase diagram. The phase diagram now being in terms of the control parameters of the system, being the total density that you have, and the ratio of the diffusion coefficients. And then you see, like in a liquid-liquid phase separation, you have a kind of binodal and a spinodal. This orange area is linearly unstable, and the blue area that you see needs a finite external perturbation for the pattern-forming process to start with. So let me summarize this part of the conceptual model. So the main point I want to make is you can turn equations into a graphical construction and a geometric classification of these pattern-forming systems. What have we learned? We have learned that we can answer these two basic questions. Why do protein patterns form? And what determines the patterns or the properties of that pattern? The answer for the simple case is that the slope has to be negative. But if you look at a multidimensional component system, which I don't have time to talk about, you can actually give a full geometric classification of these instabilities in terms of geometric properties of the manifold of equilibria that you have in a multi-component system. What determines the properties of the patterns? You saw it's this interplay between the local reactive equilibrium that pulled the system towards the nucleus and the diffusive exchange of proteins between these different compartments. And again, this can be worked out in a quite complete, what we call a local equilibrium theory that explains pattern-forming systems with mass conservation in quite general terms. Now that we have understood that, let me ask this daring question of how molecular is really molecular, going back now to the real biological system and trying to understand not just a conceptual model, but the actual biological system. So here again, you have this reaction network. I talked about it. You can turn it into a diffusion reaction set of equations with all the boundary conditions and work it out in terms of finite element simulations. You recapitulate the oscillations that you see in the living system. There's one very strong prediction of that model that you may be able to intuitively guess. Namely, the min E concentration has to be lower than the min D concentration. This means, right, if min E is higher than the min D concentration, it's the antagonist, so it kicks off the min D proteins right away, and they don't have a chance to form a pattern to begin with. So this antagonist has to be weaker than the min D protein such that patterns can actually form. So that's a prediction that you make and that you can test experimentally. And it was tested by a very ingenious experimental setup that Petra Schwille came up some time ago. And what she did is she took the min proteins out of the E. coli cell, put them into a in vitro reconstitution assay where you have a membrane on the bottom and just on fluid on top, which has physiological concentrations. And then you put in just the two proteins, min D and min E, and also ATP, of course. So you have a system fuel, but nothing else. So you're sure that you actually do have just the two proteins we're talking about. And then that's what she saw. This was Martins Lose working in her group. They saw these beautiful spiral patterns. And that was the first reconstitution of these biological pattern-forming systems in a controlled fashion. And that you can use to check on the theory. And that's what they did. So they checked experimentally what happens as you vary the ratio of the two proteins. And remember, I told you our prediction was that the concentration of min E has to be less than min D for patterns to form. That's not true. It's just wrong, right? So if you look at this experimental data, there's a very broad range of concentration ratios where you have pattern formation that extend way beyond what is predicted theoretically. Now, well, this makes you think. And then, we really had to go into the biochemistry literature and ask ourselves, what are the basic biomolecular mechanisms giving rise to the interaction between those two proteins? I alluded also already to the min protein, min D protein itself as an ATPase. And what's there in the literature, which is rather indirect evidence, is saying that the min E is also a switch. So it exists in a closed conformation and an open conformation. The closed conformation cannot bind to the membrane. The open one has the membrane targeting sequence out and can bind to the membrane. And what there was evidence for is that the min D protein makes the min E open up. So it's able to be binding competent and then binds to the membrane. Now, to make a long story short, Jonas Denk, a PhD student in the group, extended the standard model of this min system to include that switch. And what it turned out is that this latent min E state is really the key to understand stability in that system and fully covered the regime that they saw experimentally. Basically, what it does is that it can work on a higher min E concentration because min E is only recruited, activated when needed, and when not needed, it stays in the latent state. So it acts like a buffer, like a reservoir for the min E concentration. Now you can test the theory once more by asking your experimental colleagues to make a mutation. So Simon Kretschmer in the group of Petra did that. He created a mutant of the min E, which doesn't have the switch. And indeed, when he redid his experiments for the system that was lacking a switch, it recovered the original prediction of the concentration range being shifted to way below 1. So this means you have an explanation of the in vitro data. Now, what about in vivo? What about living systems? A real E. coli cell, as we have it, can you make a phase diagram? Can you vary min E and min D concentration, the control parameters, to check the theory? And for that, I was chatting with Suk-Yun Yoon for a very long time. So this whole topic, this whole project that we had together was going on for many, many years. And Suk-Yun Yoon and his group, they succeeded in creating an experimental system where in the living E. coli cell, they could change the concentration of the protein levels by some gene engineering that I don't have time to allude to. And finally, the data came, and this is what they saw. And on the right-hand side, you see what the theory says. So on the left-hand side, you see, as a function of the two protein concentration over a range, say a decade or so, roughly 2 decades maybe, that the regime where you see pattern, the colored region, is very broad, meaning you recover also in the living system this feature of robustness that you see in the reconstituted system. And the same parameter regime, this is not in non-dimensional units, it's actually in dimensional units, we find with the theory that this matches very nicely with the experimental data. It's not just the regime that matches. If you look at the chymographs that's shown here on the right and the left, there's also a match between the type of patterns, traveling waves versus standing waves, so the green region versus the blue region. And if you look into more details, even the wavelength, the value of the wavelength that we find in the theory matches with the experimental data. And as you can imagine, I was very happy to see this experimental data because for us over many, many years, this was really a final result where we could show that these conceptual models to more realistic models are able to explain a feature that you find in a living system. So that means you have this fully quantitative model and it's biologically relevant, but now you might ask at the end, is there novel physics? Or was this just trying to explain interesting biology? And that's the last point I want to make in my talk, namely that there is new physics and there are new laws when you look at these protein pattern forming systems. And I would like to illustrate this with yet another example, but you don't have to know much about the example. Just look now at the movies where you see a polar pattern forming in a model for C. elegans, and notes the difference between the prolate and oblate shape and how the interface actually forms. So you see an interface forming, and then after the interface is formed, it reorients in the prolate and the oblate shape. And as you saw, it reorients such that the length between the blue and the red regions, so the interface length, is minimized. And that is very much like a surface tension, where you also have a feature that minimizes length. But you have to note, right, a diffusion reaction system is a gas, basically. There's chemical reactions, but there's no pairwise interactions between particles that you put into the theory, just the reactions forming dimers or binding states, but not pairwise reactions that usually give rise to a surface tension. So from thermodynamics, statistical physics, there's a famous relation for the formation of interfaces, their characterization, and the concept of tension. It's the Gibbs-Thompson relation, and it says that if there's a curvature in an interface, the interface tension will drive it back to a straight form, and there's a relation between the curvature, the interface tension, and the jump in density delta rho from the liquid to the gas phase, and the change delta mu of the chemical potential that you have across between the two regions that you see here. So basically, this chemical potential difference creates a gradient, and this gradient gives rise to a flow, a particle flux, and that evens out the concentration gradients and pulls the interface straight. So that's the thermodynamic picture of surface tension and the Gibbs-Thompson relation. And now we ask ourselves, is there an analogous concept of surface tension in pattern-forming systems, despite the fact that we don't rely here on forces, but reactions and non-equilibrium currents? And can those systems then show the same kind of behavior as the corresponding mixtures in equilibrium, like foam-like structures, for example? So let me explain in a hand-wavy way. And behind that, there's a singular perturbation theory that's in the paper that we wrote. So imagine or remember that I emphasized at the very beginning of my talk that there are cyclic fluxes between the membrane and the cytosol and between different states. And that's visualized here. So at the interface between a high and low density region on the membrane, that interface defines a circular current that goes with it. And now let's look on a projection, so looking on top of that interface. And here it is, so it's a top view of the interface. And this narrow band that you see is the interface, the middle line being the inflection point, and the red region, the zone of detachment of proteins from the membrane to the cytosol, and the blue region that you see, the region of attachment on the high density end from the cytosol to the membrane. And now just look at this geometrically. As you have a curved interface, simple geometry tells you that the outer part of an interface that's bent outward is stretched, and the inner part is compressed, as you see in this picture. And the opposite is the case if you bend downward. Now the outer part is compressed and the inner part is stretched. So what you do is by bending the interface, you create an imbalance between the attachment and the detachment currents. So there will be a surplus of proteins in the cytosol on the left, versus the right, because of this imbalance of attachment and detachment fluxes. But since you now have a density gradient, there's Fick's law, and this Fick's law will transport particles from the upward bend region to the downward bend region, and that fills up the surplus of proteins, evening out the concentration and giving rise to a flattening. So you can put this in formulas, the formula saying that there's a concentration gradient, which is giving rise to a surface tension, very much like in the Gibbs-Thompson relation. The reason being, though, that this is non-equilibrium transport currents giving rise to surface tension and not pairwise molecular interactions. So now, is there a relevance of that surface tension? So we look back into the literature. This is older data from Petra Schwille's group, where Petra Schwille and her student, Philipp Glock, they were looking into a different parameter regime of min protein patterns on the surface. And what they saw is this, right? This picture here. And it looks like a foam, right? It has these features that you typically expect from a foam. So let's check. And we checked. So if you put here tripods at the vertices, you'll find there is Plato's law. There's a 120-degree angle, pretty precise and pretty narrow, just as you have it in two-dimensional liquid foams. If you look at the size distribution, that's different than in foams. This type of foam doesn't coarsen. It develops an equilibrium steady state of surface area that's given here by the distribution that you see on these data. Now, since we have a quantitative theory, what we did then next is to say, well, then let's use this quantitative theory of the protein patterns and study what they have here on the computer to see what the phenomena are that give rise to the foam-like patterns that you see, and whether we could identify even more foam laws, mesoscopic laws, in these protein patterns. So the first thing Hendrik Meyer did, the PhD student in the group, is to say, let's start from an equilibrium structure that you see on the left, and then let's compress this equilibrium structure down so it's no longer equilibrated, but it has a cell size which deviates from the equilibrium size. It's too small. Then as you let the system run, it should relax back to the equilibrium state. And you should note here on the right, there are vertices and there are cells with 5 edges, with 6 edges, with 7 edges. And as the dynamics unfolds, they either grow or shrink or stay constant. And what you see is this. And if you translate that into a quantitative plot, what you saw there is that the cells with 5 edges, they grow. They actually shrink. The cells with the 7 edges, they grow. And the cells with the 6 edges, they stay constant. That is the celebrated von Neumann law that you also find in liquid foams. And in a way, it has to be there because the von Neumann law is simply based on the fact that you have a surface tension, which we have, and the remainder of the law is based on the Gauss-Bonnet theorem, which you work out here for this foam-like geometry, and then you find that this von Neumann law must be valid based on geometry alone and the fact of existence of a surface tension. Why is there a finite equilibrium size? So why doesn't this coarsening go to completion and the system dries out? The reason is that if you do the reverse experiment where you scale up the system, so you make larger sizes, you get reverse transitions where you see here in the movie, you have kind of cell divisions that happen, so that if cells grow too large, there are events that cut the cell size by, as I said, cell-like division events.
Just to let you know, you told us to warn you when you're at about 45.
Yeah, so I'm perfectly. Thank you very much. So let me, at the end, skip that and go back to the data. So when we showed this to Petra, she was like, oh, we have some old movies that we never published. We want to share them with you. And these are the movies, and they exactly see what we saw in the simulations. The 5s shrink, and you have these cell division events here on the right-hand side. All right, so to finish, maybe just one more short thing which kept me busy over the last years or so, namely the question whether we can use all that to build lifelike systems from the bottom up. And I think that we can do that on the basis of theory, not just on the basis of experimental data. Why? Well, because as I explained to you, in these cell-like systems, you have all the ingredients you need. You have a membrane, you have proteins, and they create mechanics and they create geometry. The membrane and the proteins interact in a way that the proteins can sense the geometry because they can read off the geometry. And the proteins can also change the geometry because by binding to the lipid vesicle, they can create spontaneous Gaussian curvature. So in that way, you have a feedback between geometry and mechanics, and that gives rise to this phenomena I started my talk with, namely to this cell-like motion of vesicles, which you can also recapitulate in a phase field model on the lower left. That's done by Jan Willeke in my group. So let me close with a final conclusion slide. What happened? So I hope you can still see it. Can you see it?
Not yet. You appear not to be sharing.
I think I need to share again. Sorry, I dropped you out of the share. Now you should see it, right? So my conclusion is that these mass-conserving diffusion reaction systems have led us to rethink how we consider pattern formation and get more physics into the theory of pattern formation by formulating a local equilibrium theory based on mass redistribution. And I hope I could show you that these protein pattern forming systems help us to understand in vivo living systems. They give rise to new kinds of mesoscopic non-equilibrium phenomena, which we call Turing foams. And they also can help us explain cell-like motion. Where do we go from here? Well, I think one of the great frontiers is to combine dynamic manifolds like the starfish oocyte that you saw in one of the first movies and hemomechanical coupling to start to understand the interplay between membrane physics, geometry, and pattern formation. And as I tried to allude at the very end, this is a very exciting time to see how far we can go in creating bottom-up systems with lifelike properties that we can control and maybe at some time help constructing synthetic or lifelike cell-like systems. Let me thank at the end the people who did the work. So there's a range of PhD students that have been doing a lot of the work. Maybe I single out Hendrik as the one who did the work on foams. He just finished his PhD thesis. Spectacular student. He's now with Boris Schreiman starting his postdoc. And below you see Petra, Nikta, Kes, Sukyun, and Lidevai, experimental collaboration partners. And it's always fun to work with them because they connect us with reality and keep us honest in the way we do theory. So thanks for your attention.
I guess we are wide open for questions. I see one. Cynthia, I think I'm going to find and try to get you to talk, to speak directly. Let me see if I can unmute you extremely quickly. Sorry, it's my first time trying to do this. Okay, why don't you go ahead and talk?
Can you hear me?
Yes.
Okay. So I wanted to ask about the affective disorder. So if you had a point-like disorder, would the system just screen that and not care? If you had more line-like or 2D disorder, would it have to conform to that? And what if you had disorder that was actually a third chemical reaction? that was inserting itself at certain localized points.
Okay, so disorder is of course an important feature, and it's everywhere, because when you think about the system, it's a cytosol, and the cytosol is not just a homogeneous fluid. There's a lot of stuff that is there. And what this does is basically it modifies the diffusion coefficient, and it doesn't really matter that much, you know, how large the diffusion constant is in detail in all these different regions. If I were to modify that a bit here and there, this would not change the qualitative outcome. Of course, the quantitative curves that you get, they are shifted. What we also did is we changed on the membrane in a line-like fashion, so a patch-like fashion, the attachment rates. So because we wanted to know whether patterning of attachment and detachment rates along the surface of such a system has an effect. And the short answer is, if you do it weekly, it doesn't matter much. You have to make it quite strong to change the pattern that you see. What matters most is actually the geometry. I couldn't show that, but if you put the pattern into a wedge, or you change the external geometry by modulating it in a random way, then the pattern changed quite a lot, because then you change the penetration depth of the patterns in the cytosol, and that has a strong effect on the pattern that you see.
Great. I don't see any other hands up, so well, I guess I'll dive in and ask a question myself, if that's okay. So you early in the talk, you mentioned, you know, you talked about how a lot of this was driven by the ATP energy transition. And I thought that was really interesting to wonder about whether we could spot some of these features of dynamics or structure by focusing on energy consumption patterns or signatures. So that's, I guess, a long and complicated way of saying, do you think energy dynamics would show similar timescales or give us a way of looking at these issues?
So there's a few facets to that, and to answering this. ATP in a cell is abundant, and so it is here. So there is no shortage in terms of the fuel that you have, and it doesn't really run out. The other question that you might well ask is, you know, where is most of the energy consumption happening? Is this at the interfaces? Is this on the boundary, in the bulk, etc.? We didn't look at this yet, but I think it's quite an interesting question to ask, of where the dissipation, the entropy production in the system actually happens, and most of the energy is consumed. My bet is at the boundaries, because there is where the non-equilibrium currents actually take place, and they are the hallmark of the non-equilibrium nature of the patterns that we see.
Great, thank you. Shima, you should go ahead if you have a question.
Hi, this is Shima. I have a question about infinitely making the diffusion large, or like in terms of like in interfacial tension, if the interfacial tension is extremely small, so the interface can stretch a lot if the interfacial tension is very small. What would be like in this system where there is a reaction and the two components have to diffuse?
So how can you tune the interface tension? That's basically your question, if I understand it correctly.
Yes.
In many interesting ways, because the interface tension has its origin in diffusion and reaction. Diffusion constant, of course, has an influence, because it sets a scale, and all the reaction rates that you have in the system. So for example, if you make up certain pattern-forming systems, like 2 or 3 or 4 component systems, what you can do is by tuning the rates and tuning the diffusion constants, you can get very particular patterns in this multi-component system. You, for example, can tune the parameters rates here in this case such that you get phenomena like complete wetting of one type of species surrounding the other one. So it's quite a simple way of tuning the shape and morphology of multi-component pattern-forming mixtures, since the reaction rates are the control parameters.
Thank you.
Great. I guess, Doug, you should go ahead.
Sure, thank you. Thanks, Erwin. This is nice. I wanted to ask you about the Turing foams and the rate of change of the areas. So by comparison with the von Neumann law, well, the von Neumann law says that the 7-sided should grow and the 5-sided should shrink with exactly opposite rates.
Yeah, that's the good point. Yeah.
Yeah, in your case, the 5-sided shrinks a lot faster.
Yeah. So that's a feature which is not the same. And to understand that, there is no simple answer that I can give you. The only answer I can give you is this has to be worked out in detail. So just to give you a glimpse of what one does is one creates an interface theory. So one considers an interface, one writes down the diffusion reaction equation and maps them onto an interface by doing what's called a singular perturbation theory or a asymptotic matching analysis. And then out of that, you get things which are like Stefan's law, and that then helps you to formalize the actual dynamics of the interface in terms of the dynamics of the pattern-forming systems. And that's a quite lengthy calculation. And as said, there is no real simple answer to what you just asked me. But we can calculate the difference, and it's quantitative.
All right. So the mapping, though, onto von Neumann seems maybe suspicious then. What about maybe other size cells, other side numbers? 8-sided, 3-sided?
We didn't see them.
They just don't happen. Okay.
In the system size that we study, they don't happen. And doing simulations for larger systems is a bit challenging. So they could be there, they just may be super rare.
Right.
And then we don't see them in a finite size system.
All right. Thank you.
Welcome.
I think there are a couple of questions in the Q&A. One says, is it possible to say something about the robustness of such mass-conserved patterns if we imagine the existence of advective flows in the bulk, e.g. contractile flows?
Yeah, good question. We looked into that. We have a work with K.S. Decker where he looked at a microfluidic chamber where he put by purpose a flow in the cytosol or the 3D bulk region, and it has an effect on pattern formation. A bit of a counterintuitive effect because the pattern reverses in the direction of the flow. So the effect is strong, and the reason is that these patterns penetrate in the bulk. So as you apply a flow, of course, you advect the components in the cytosol, and that feeds back into the pattern on the membrane. And if you write me an email, I can point you to the paper.
There's one last question on the chat, which is, having looked into these mechanisms for pattern formation in biological systems, do you have any insight on how these could have initially evolved in early cellular life?
That's always a hard question because it's very hard to imagine what happened during evolution. I'll give you the following answer. We have a paper with Ludwig-Maximilians from the TU Delft. And what Ludwig did, she looked at the CDC42 system, which is the one that shows pattern formation on yeast, and which has this very, very complicated network. And what Ludwig then did in her experiment is to delete specifically one of the most important proteins that you could imagine that gives rise to pattern formation in yeast. The nice thing about yeast is that it seems a way out. So after 1,000 generations, this system recovered function completely. And they could look, because they had all the strains available, they could look what happened kind of backward in evolution. And they saw that this initial mutation they put in was followed by another set of deletion mutations, so that you, in the end of this cell biology evolution experiment, you had a reduced network of proteins. And interestingly, this reduced network of proteins in the yeast system has the same kind of wiring and topology as the MIN system. And that's surprising, because these two systems are not related evolutionary, right? The proteins have very different origin. So what this points out to me is that it's not so much about phylogenetic trees to understand how these things evolve, but about basic physics mechanisms that give rise to the most simplest pattern forming system that one can imagine. And I would say this protein system with min D and min E is pretty minimal. It just has two components, two species, not more than that. More recently, what we do, and others do as well, is to say maybe one can get pattern formation not out of proteins, but out of RNA and DNA, if you make RNA and DNA interact in a very specific way. There's no conclusive answer yet, but my suspicion is, and my hope is, that even with RNA and DNA and the interactions they can have, you can create systems that show pattern formation with these kind of macromolecules. And that's, I guess, as far as we can look back in evolution to figure out of how these pattern forming systems may have evolved.
Great. Thank you. Madam, do you think we can squeeze in one more question? Maybe Cynthia has her hand raised. I should go ahead, please. Sorry, Cynthia.
No, I think I clicked that by mistake, but I can ask one part of my question that you didn't get to, which is if you had a third chemical reaction, would that, and it was in the only localized spots, could that destabilize the pattern or cause a localization of one of the domain walls?
So you want to link another reaction to the reactions that I already have.
Right.
Of course it will, because the way you can think about this is that this would then trigger off the production, say, of one of the key proteins, and thereby go through the phase space of the pattern forming system and create bifurcations via the second reaction or the third reaction that you have. So for sure, as you make things broader, there's more room for control. Yeah, and one example, maybe I show this, so just as my last thingy here. If you look at this, this is maybe something similar. Here, what you do is you have a reaction network, and you couple this reaction network to the actin cytoskeleton, and by the reaction network, you activate the myosin proteins that then create stresses and strains. And what we did here with NICTA is to light activate proteins externally such that they give rise to spatial patterns that make this cell into a square instead of a sphere that it was. So yes, I mean, there's a lot of room in linking different parts of reaction schemes to gain control over cellular systems.
Maybe this is a good moment to thank our speaker for an excellent talk. If he's willing to hang on for a few minutes, maybe
Sure.
there'll be further questions, but uh, let's first thank him before the audience leaves. Thank you very much. This was really lovely.
Yeah, thanks for having me. I enjoyed it very much.
We
Prof.
actually do have one person with a question. Maybe Karthik, are you there? You could unmute and ask your question while you're there, since we're hanging out now.
Oh yeah, thank you. I don't know much about non-equilibrium physics, but I just wanted to ask, like, I've seen talks where people talk about entropy production rates and like breaking of detail balance. Is that in some way that you can see your systems in this sort of ideas?
You could, but that's not really what we do. What we do is we say there is broken detail balance and we work out the consequences of that because our chemical reaction network breaks the detail balance. What we don't do is, and that's quite some work, to translate this in a thermodynamic consistent framework where you would need up on top of what we have, the delta mu's, the chemical potential differences, and then construct a theory that allows you to write down the entropy production rate of the pattern forming system. There's work by Esposito and others. They have done this for simpler systems, so you can do it, but that's not what we did. And having said this, if you want to measure that, it's very hard, right? If you say now, once we have calculated the entropy production rate, so the heat basically in these systems, you need a way to measure the heat. And that's a super challenge because you need to design an instrument that is able to measure these minute amounts of heat in the system. I know that Dan Needleman at some point tried to do that. I don't know how far he got, but that's a challenge in the field to be able to measure that. And once people are able to measure it, I think I would jump on the wagon and try to calculate that. Otherwise, I would shy away a bit because it's quite complicated to write down those equations.
I see. Thank you.
May I quickly ask a kind of specific question? You told us about this mechanism for the surface tension, right? It has, there's a detachment zone and attachment zone. It seems like it really depends on those layer thicknesses, right? The strength of this.
Yeah.
So those have to be, since this is such a long wavelength phenomenon, those have to be not so small, right?
Those interface widths are actually pretty small.
But then how do you get the effect of this differential length?
Yeah, so it should be not too tiny because otherwise you have no effect. It shouldn't be too large because otherwise you wash away the structure and you will get a mixture-like behavior and a foam-like behavior. So we really need the length scale of the cell and the length scale of the interface thickness. So if you look at the data that are produced by Petra or look at the data that we produce with our simulations, there's a factor of roughly 10 difference between the cell size and the interface thickness.
I see. So there's sufficient scale separation for it to be a surface thing, and there's it's not so small that these things are, a modest curvature. They can influence fairly long scale curvature. So there's some information in just looking at the interface fluctuations that help you in some way understand some underlying biological noise in this thing, or is the interpretable information there in the fluctuations?
So noise in biology is mostly disorder because there's membrane disorder since you have different lipids. There's disorder in the cytosol because there's crowding. So that's the main source of disorder. When you ask about fluctuations coming from demographic noise because you have chemical reactions, the point is these proteins are about 1,000 typically in number. So square root of 1,000 is a pretty small number. And that means number fluctuations are kind of small. And you don't have to really worry about that so much. Thermal fluctuations don't really play a big role beyond the fact that diffusion, of course, is based on entropy and thermal fluctuations. So the answer is basically fluctuations are subordinate to the attractors that you find from the nonlinear dynamics and the reactions in the system.
I see. I see.
Yeah.
Thank you. We're at 15 minutes past the hour. Maybe this is a good time to thank our speaker again for a fascinating talk. Thank you.
Thank you.
Thank you.
Thank you for having me. It was really fun. Thanks a lot.
Pleasure.
Thanks, Erwin.
Thank you.
Thank you. See you.
Thanks again. Take care.
Yeah. Bye-bye.
Bye.
Bye all.